proper relationships where things went far more smoothly. But the effort taken to develop that understanding upfront was pretty important. For our afternoon, we've now got 2 speakers lined up for you, and after 2 speakers, we're going to have a panel discussion. So you have an opportunity now. You've got the wealth of expertise that the meeting has lined up for you. It's your opportunity to ask them more strategically questions that are related to successful partnering, successful outsourcing. So I'd like you to think about that as we go through the afternoon. But in the meantime, what I'll do is I'll invite Gavin Bennett from Cellzone to take the podium, and I'll ask Gavin and Paul eventually to just give a 1-minute presentation, introduction to themselves, so that you can understand where they come from and their candidacy for their role in this meeting today. Thank you.
So, thanks, David, and thanks for the meeting organisers for inviting me. I'll start with a little disclaimer. So I know this is an RSC day. I'm not a chemist. I started a chemistry degree and was then tempted away to the dark side of biology, and I'm a pharmacologist. So I now work for Cellzone. I did followed up my degree with a PhD in inflammation and pain, then went on to work for Rhône-Poulenc-Rora up to their merger, then worked for Celltech in UCB following that. Now work for Cellzone, where I look after the in vivo work and the pharmacology work, which is generally all done through outsourcing. So what I'd like to do today is to talk you through a little bit of how we work and some of our experiences in terms of outsourcing and the difficulties and the challenges of managing some of those relationships. So I guess from our perspective, we're a sort of mid-sized biotech company, about 100 people, split between our site just outside Cambridge and a site just outside Heidelberg in Germany. So I'm thinking about how biotechs fit into the sort of drug discovery world. You know, if we think about the traditional pharma company model, running everything from basic biology through discovery, development, marketing, sales, production, right through. Most biotechs have tended to focus on small niche areas within this. You know, whether they're platform-based biotechs working in a very small area, diagnostic biomarker ones, you know, working more within clinical and clinical development, or whether, you know, they're sort of broader drug development partnerships, such as the kind of things that we do. And obviously within this, there can be a significant amount of difference between, you know, how basic the biology is, whether you're really looking to discover new areas or whether you're looking to pick up validated targets and take them through, and also the extent to which the companies build through, you know, late discovery into preclinical and clinical development. But effectively, most biotechs fulfill really discrete functions in there. The internal resource is focused on very key aspects of what they do, but they're not going to have the resource to be able to take things right through. Now, obviously, there are significant historical examples, the Amgens, the Genentechs, that have managed to build from being a small company to, you know, fulfill all of these functions. But, you know, certainly in the current climate, there are a few biotech companies that have either, you know, the aspiration or certainly the ability to fulfill all of the kind of functions that they need amongst themselves. So, you know, we all tend to rely on conducting work externally with academics or with CROs. And obviously at the extreme, you can have virtual companies which manage programs entirely through partners. So what are the opportunities and challenges that offers? Well, you know, for us, one of the key ones was it allows us to focus on this core competence, allows us to really build our expertise and make sure that what we do best, you know, we can do to the best of our ability, really take ownership of that. It allows us to access specialist expertise, you know, actually get the best people to do those functions that we want, rather than simply the best people that we could employ at the time. That allows us to be much more dynamic, lower fixed costs, and focus our spending much more minute by minute. You know, we do have the challenges. It doesn't allow us to build the internal expertise. We're accessing externally. We need to do that again each time. And particularly for, you know, smaller companies, it's very difficult to select and control the kind of expertise that you want. You know, what we do find is it's difficult to access that expertise in a timely fashion and with the quality that we're after. And there are aspects of coordination as well. So, you know, most biotechs operate this mix of internal and external. Proportions vary from company to company, but they'll also vary in time depending on the life cycle of the project and on the company itself. So this is the way we work. So our internal effort is focused on our core business, which is our chemoproteomics platform, and this allows us using mass spec to look at the interactions of compounds, even biologicals, with key targets. And we do that in a natural cell environment, so within cell lysates or within blood, within tissues. And that underpins all of the work that we do. We've applied this to the kinase field, where basically we can run selectivity assays across the entire kinome, and we've also applied it to multiple other structural target classes. We then focus on the in vitro biology that takes us from that initial chemoproteomics, affinity measures, and the medicinal chemistry to support the development of compounds focused on these. And that's really where our main internal focus is. Obviously, we have some other supporting areas, but we use external partners to provide all of these other specialist functions that we need. The PK/PD, process chemistry, non-clinical, clinical efficacy, and formulation work. But we also use external partners for additional capacity where we need those. So we've ended up, with two different approaches to working with external partners. The first is outsourcing effectively a single task, and this is very much a sort of we'll tell them what to do kind of idea. And effectively, the CRO takes on a defined task that we determine and control. The intellectual input, though, comes from our end. And effectively, what we're buying here is time, both human time and machine time, consumable. At the other extreme, there's this, outsourcing a project, and this really involves us, passing a single thing over to a CRO who has significant expertise. Tell us how to do this best. We need, obviously, work up front with the CRO to define the task, work through what the process is, what the successes are. But ultimately, a lot of what we're buying in this is the expertise and the experience from the company. Now, we use a balance of these for different things. Particularly, the task outsourcing we tend to do less, and this tends to be areas where, we're looking for additional capacity. So we're looking short term to build extra hands and get extra time in there. So I've kind of looked at this as an analogy in terms of exploring for oil, similar to pharmaceuticals in the sense that we spend an awful lot of time and a very technical and expensive process in exploring for things, very high attrition rate with a high value product out of the end. And effectively, in terms of outsourcing a task, the analogy is effectively engaging a team of drilling engineers, us telling them where to go, what to do, they go off and do it. In terms of outsourcing a project, we engage an experienced team who know what to do. They go off and come back according to the success criteria and tell us how we've got on. So I've got a number of examples here related to the kind of things that we've outsourced. They're not particularly focused on chemistry, as I explained from the beginning, but I think there's some key learnings from these that apply across the board. The first of these is in Vigo PK. And this is one of the first areas in which we're really involved outsourcing. And it's a really critical function for us in selecting and developing those small molecules. And there's key things we need from it. We need access to these models on a really timely basis. We need to be able to get the data returned quickly, but we also need to know that that data is of a really good quality. Now, notionally, there's a large marketplace out there. There's many suppliers, established methodologies, very standard formats. So, this should be, an ideal opportunity. We can pick and choose multiple suppliers, and everything should look the same. Our experience is very different. So, this is an example of a single compound that we ran as a simple IVPO rat PK study of 4 different contract research companies. We used the same compound, the same dose, the same vehicle, the same timings, and the studies were identical. What you can see just from the graph is the extent of variation that we get back. Significant different outputs, both in terms of the final generated figures and the quality of the data. What we've got here, reiterated in the graph here, is a measured clearance ranging anywhere from 1/3 of liver blood flow up to about 1.5 times liver blood flow for the same compound. Now, clearly, our ability to make decisions based on this data is quite compromised. What you'll also see, though, if you look down the bottom row, is just the time that some of these things take and the variation in this. Supplier 1 here was able to turn around data within 12 days, whereas it took 51 days from initiation to receiving data from supplier 4. Well, to be honest, the kind of business we're in, by the time it's 51 days in, we've lost interest in that compound. We need to know far faster than that. So, this has been a bit of a bugbear of ours in this particular area. We've seen a wide variation across suppliers. Now, we're lucky in this sense, in this area, in that we've got quite a lot of internal expertise. We've got a number of fairly senior people who've worked in this area, and we've been able to interrogate data, go back, deal with all of those quality issues, and actually come to the bottom of what those issues were. But, it's taken a huge amount of time from us in terms of auditing facilities, auditing procedures, and just day-to-day going through data. And that, results in us being able to refine work with CROs and get things up to the kind of quality that we want to get to. But it takes an awful lot of work to get there, and a really significant overhead to maintain that and keep it from slipping. And so, although we started off considering this as outsourcing a project and really using expertise that existed within the CROs, it's felt much more like outsourcing a task and really just getting, handle turning done. So next example is some formulation work. Now, we'd identified a lead molecule, demonstrated efficacy in a key disease model. However, as often happens with these, the preclinical PK was limiting. So, most exciting compound inevitably is one of the hardest to work with. Exposure is variable, and the standard formulations pretty difficult to work with. So, we had discussions with a broad range of CROs regarding formulating for this material. The proposal for the initial work is broadly on the same lines from most of the CROs that we dealt with. The variation in response was really around timelines and cost, but also the depth of the expertise, the ability to actually apply that. A number of suppliers that we spoke to were able to technically go through and do the aspects of work that we wanted to do, but we're also looking here for expertise and for guidance. So, we went through the work package, evaluated a range of formulations, and selected a new formulation. We were able to produce much more predictable PK, significantly less variability in either the red or the blue formulation lines than in the original black curve. We were then able to subsequently use this in long-term studies. So we've been back and kind of worked through what it was that made that such a success for us. And really, it's not so much the technical process we've been through, it was the responsiveness of the CRO and the can-do attitude for these. You know, inevitably, we always want to do things tomorrow. You know, we've got a study, we want to get it in immediately. The ability of the CRO to be able to take on challenging timelines and to be honest with us about when they could and couldn't deliver them, it was vitally important. And then the expertise element of not just being able to conduct this study, but being able to place it in context and to look at, you know, to advise us on the next moves, how we then build that on to go into to further studies. And that gives, you know, for us, there's a much higher likelihood of repeat and follow-on business from the experience we've had there. And the third example we're going to go through is some work in terms of in vivo efficacy, so the things closest to my background. Inevitably, what we're asking in these studies is how efficacious is our compound? Now, that's a pretty simple one to answer in general. But underlying it, what we really need in terms of moving these compounds forward is to understand how they achieve efficacy. You know, in general, the models that we go into in terms of preclinical animal models are fairly contrived. We want to understand how this is going to work in the clinic. So we have a number of programs targeting adaptive immunity, and we've used for this a delayed type hypersensitivity model. It's a pretty standard mechanistic model related to T-cell reactivity. And we've identified, you know, over 10 CROs that were offering the model. Comparable prices, comparable timelines, some variation in the formats of the model, but broadly similar. But the experience that they offered of the model was pretty variable. Few of them ran the model on a regular basis. Most were able to show us some kind of sample data, but very rarely of a quality or of, you know, appropriate pharmacology for the kind of things we were after. So this is simply a sample of some data that we ran with one of the CROs. So I put up earlier 2 key questions. How efficacious is our compound? Well, every one of the CROs we went to could have told us that. The one that we really want to know is how it achieves the efficacy. And out of, you know, that sort of 10 to 12 CROs, there were only 1 or 2 of them that had enough knowledge around the model and also the ability to build things on and to interrogate further to help us understand how this achieves efficacy. And that's the thing that, you know, gives us the value. So what we've done in the example of this model, but also, you know, more profoundly through our biology models, is to focus on CROs which have a much broader experience of these models, experience of multiple compounds, understand the underlying immunology. And that allows us to then, you know, add on extra work in there to build up our understanding around the compounds and to link it better to our clinical plans following on. You know, in order to make this work, though, this does mean, you know, we have to share internal data in there. You know, a lot of the work that we do up front feeds into the kind of questions that we'd like to answer with this. So it becomes much more of a collaborative initiative. So it's about working smarter with that CRO, but ultimately gives us a lot greater value. So I've touched on expertise a number of times, and I think, you know, whenever we meet with CROs and talk about doing a particular study, expertise is always one of those key things in the initial pitch. But what we do find is that there is an underlying expertise which varies a lot between companies that are effectively offering assays or processes versus the companies that actually understand those and have, you know, taken the time, taken the ownership behind those. Now, there are times when all we need is the process, but in general, there's a lot more value for us in being able to place that into context. You know, and keep saying it's about ownership there. So we need expert input, not just in terms of this ownership, but to make sure that there's rigorous analysis of the data in terms of the PK examples earlier on a number of the occasions where that data is variable, it's simply because of not following rigorous procedures. We need to place that data in context so that we can really understand where we take it. And that differentiates all of the CROs for our project. So what it does mean, and what's come through from, I guess, each of those examples, is the amount of internal overhead that we need to commit to running these studies. Now, from a CEO perspective, from a board perspective, outsourcing is normally seen as a way of getting this work out without there being an impact. What we've shown on each occasion is that we really need to commit significant internal resource to support this. It can simply be administrative in terms of getting compounds to and from, and particularly in terms of, for outsourcing process work, dealing with data on a day-by-day basis. But in general, it's expert support. And it's about needing to have people with enough knowledge to be able to understand the study types, to be able to select them in the first place, and then to deal with the studies as they're ongoing. Most studies that we're going to run, there isn't going to be a single outcome at the end. There'll be multiple decision points in there, so there'll be a lot of need to have experts involved in there and provide the context for decision making at the end. So we really need to invest in these partnerships, ensuring that the studies are run in the best way to suit us and also our partners, and effectively get the studies run in the way that we would run them ourselves if we were resourcing them internally. But that means a significant input for one-off studies. And this internal overhead always gets grossly underestimated. Now, this isn't a one-way process, because clearly, as I said earlier, we're 100 people. We're not going to be able to take programs through from early discovery right through to be able to get them onto the market. We develop programs in collaboration with big pharma companies. The partnerships are varying degrees of involvement with those companies, but they do have a significant influence on some of these decisions that we make. So, we're often influenced by the experience or the bias of the partner, the extent to which different types of data are important to them. And it can be technical. It can be things about how assays are run and how data is presented. Things like time-dependent CYP inhibition. We have a number of partners who have effectively decision trees that they're very comfortable with based on distinct cutoffs. When we present data to them in different formats, it confuses things entirely, and we have to go back and rationalize all of that. In those kind of instances, it's a lot easier for us to be able to run an assay in that format and cut through to the decision making. We also, of course, get influenced by partners' experiences, both good and bad. We often get guided by them towards certain CROs, potentially away from certain CROs. And we're then placed in a certain amount of balance of having to balance our own judgments with the prejudices that our partners bring with them. And obviously, strategically, what we've seen over the last few years is the role of these sort of big pharma outsourcing groups. And again, with some of the collaborations that we have access to some of these that we can utilize to get better access to certain assays, better pricing for things, but often it's not as flexible as doing things on our own back. So there's a balance in there. So to summarize overall, I guess for all biotechs, it's at least a semi-virtual organization. Really significant functions are conducted with partner CROs, and that can be anything from small numbers of supporting functions right through to everything. There's a huge breadth of CRO offerings available, but we've certainly found a huge variation in quality and depth of experience. And ultimately, we want to try and obtain the same quality from outsourced studies as studies that we'd run internally. there's very little that we want to do that's simple and off the shelf. And I think you've seen from the first example that even the examples where things appear to be simple and off the shelf, they very rarely are. So we have to identify the best suppliers, work with them to get the best results, but we also need to balance that with making sure we have sufficient capacity and multiple options. You know, for a company of our size, the ebbs and flows are pretty great, and it's quite difficult for us to maintain enough partners where we're happy with quality and still get sufficient access to cope with our peak times. And then the last message is really not to underestimate the amount of internal work that really goes into supporting all of this external work. That's all of my slides, so I'll be happy to take any questions.
Have we got any questions from the floor then, please, for Gavin? Maybe I could ask you a question of business philosophy. So you started off within Cellzone with this balance between internal core expertise and access to external expertise, the research network. With business success, Cellzone goes on to be successful. You'd be tempted to expand that core expertise into the business, or are you happy being a virtual company in that?
I think ultimately, I think there'll be some skills that we will need to add in, but I think it's more, as I'd come back to towards the end, it's the internal expertise to support that external work. I don't, most of the kind of other functions we want to build in, we're limited by space, we're limited by multiple factors. It's not purely, we're more efficient at focusing on what we do best. And I think that's best achieved for us by getting key experts that can work externally to get those other things done.
Thank you.
You work with big pharma in collaboration on molecules. How much does that influence your decision making in terms?
In general, it doesn't. That may be a nature of the way our relationships, the current relationships that we run work, where we're very milestone driven, and so they're fairly hands off. I would say it's less influence in terms of which partners, and more an influence in terms of a philosophy and a strategy of where to get and the kind of balance between mechanism-based work and disease. So we haven't really seen negative influences in terms of partner selection. What we have been able to do is to talk to our big pharma partners about particular partner, more a case of proffering names and looking for responses than being warned off. So in general, that's been a positive thing, but sometimes we do need balance. Yeah. What prompted us was the discrepancy between the original set of data that we'd got in rat PK with some data that we then got out from efficacy models and the exposures that we saw in there. And so we followed that up, got two wildly different, and so from there it became we need to try and understand where the problem is. We've actually gone further since then. We've sent that compound to another two suppliers, and we've got another two slightly different profiles, which is profoundly worrying because, these should be fundamental. It should reflect the fundamental disposition of the compound. And, as much as you can control for different environments, different strains and all the rest of it, there is no common factor.
Disclose companies who we have.
Yes.
Could you share with us perhaps the geographic?
All over. So they encompass European companies, you know, both UK and mainland Europe, companies in China, companies in India, other companies. So there's a spread, and you know, I think it would be tempting looking at that to, you know, push it in one direction, but there is really no systematic difference either between geographical location, between size of companies, you know, any of those kind of things that you might factor into your mind at the beginning.
So does that information
Well, to a certain extent it does, but I think more than anything what it does do is give us more insight on what kind of problems we need to have our eye on day to day. And unfortunately what that's meant is a much more rigorous analysis of the root data, you know, right down to the, you know, going through traces, which, you know, obviously is a really significant amount of work that we didn't expect to be doing.
Was the physicochemical data fully characterized? It wasn't just a case of one polymorph or another that gave you a different uptake or solubility?
No, I mean, most of those were done with identical batch, you know, studies from the same pot. So, again, we've been back through a number of those variables, and we can't find any common variables that, you know.
Abin, thank you very much for an excellent overview and a really very pleasant way of getting us started this afternoon. Thank you. Christian said earlier this morning that our next speaker was originally lined up, Neil Roberts,